process of meiosis. It's a wonderful process, and that's for sure. First step in meiosis is interphase. You have the chromatin surrounded by the nuclear envelope and the centrosomes. Welcome back to Let's Learn Biology. Today we're focusing on meiosis. We have just gone through the interphase and are now going into meiosis 1. And the first step of meiosis 1 is prophase 1. Yay! First off, in between interphase and prophase 1, the chromatin, which was inside the nucleus, goes together and winds up like a slinky to become a chromosome. Inside prophase 1, there are 3 chromosomes. This is the first phase of prophase 1. In the second phase of prophase 1, the chromosomes split apart to become individual chromosomes of one singular arm-like thing. They then duplicate themselves and cross over. We now have a total of 6 chromosome arm thingies that are crossing over. These centromeres then split and divide. There's 2 pairs split across 2 and create spindles in between them. This is prophase 1. Next up on our lovely train of meiosis, we hit metaphase 1. In this phase, you have the chromosomes, which are connected on your centrosome and spindles. You also have your centromere right in the middle, which connects your 2 chromatin individuals together. In the next phase, you have anaphase 1. In this phase, your 2 chromatins are going to separate. Your centrosome then divides the 2 and pulls the individual chromatins to opposite poles. Kind of like when you're going fishing and you pull in that fish. The last phase in meiosis 1 is telophase 1 and cytokinesis. In this phase, you're going to have your whole entire cell divide into 2. This is done by a method of the cleavage furrow. Basically, it's like taking a piece of taffy, pinching it off, and breaking it into 2. You now have 2 individual cells with the whole amount of DNA in each one. Hi kids, and welcome back. To introduce the second part of meiosis, which is called meiosis 2, I'm going to have my great friend Brielle here discuss this wonderful phase for you.
First off, each cell, which is 2 different cells now, 2 daughter cells, has 3 chromosomes and 1 centrosome. The centrosome splits and creates spindles, in which the next phase, the centrosomes will travel. In metaphase 2, there's still the 2 cells which are performing the exact same tasks and functions. However, we're going to zoom in on 1 to focus. Okay, now we are in anaphase 2. The 2 centrosomes move to opposing sides of the cell, and the spindles are created. Next, the 2 sister chromatids are pushed to other ends of the cell. Our final stage in meiosis 2 is telophase and cytokinesis. In telophase and cytokinesis, the cell wall pinches inward, dividing the 2 daughter haploid cells. In the end, you will end up with 4 haploid cells. Each cell contains 3 chromosomes and a centrosome, which will duplicate itself. And for each centrosome, there are 2 centrioles within.
Yay! Thank you for joining us on Let's Learn Biology! Yay! Today, I hope you come back next week when we learn about plankton. Oh! Welcome back to Let's... You guys suck.
tubules are moved toward the center of the cell. Were you videotaping? No. There's no.
Do it again.
No. Are you taping?
No. That's gonna be awkward. I gotta say, man. Anna, you can't leave. We gotta do our video. You have to go to work.
Where do you work?
Shocked.